Hello guys, welcome back to my channel again. So in the previous lecture we have talked about the structure of bacterial cell. So in the structure of bacterial cell we have left with one portion which is called flagella. Flagella is a structure which is used by the bacteria for movement or locomotions. So basically flagella has mainly three portions or three parts, which is called the filaments, hook and the basal body. So the filament is made of a protein called flagellin, and the basal body, the hook is attached with the cell wall, and the basal body is attached with the plasma membrane. Basically the filament actually look like this. This is the filament, and after that the hook like structure which is attached on the cell wall look like this. After that, in below the cell wall, the plasma, on the plasma membrane, the basal body is there, which is on the basal structure, which is actually in the ring form. So this is the plasma membrane. So if we zoom it, or if we visualize this using the microscope, we will find a structure like this. This is the filament, and after that this is the hook which is attached on the cell wall, and the ring structure which is the part of the basal body is actually in a ring form. There are mainly four rings, and between each rings there is a rod like structure, rod, which is also called as a rod. The rings are like L ring, P ring, R ring and S ring, which is actually attached on the plasma membrane. So this is the structure of flagella, which is used by the bacteria for movement. So we are continuing with another topic, which is reproduction in bacteria. Generally reproductions are of two types. One is asexual reproductions, another one is sexual reproductions. So in the asexual reproductions there is no meiosis, that means there are no gamete formations, and in sexual reproduction there are gametes, both male and female gametes. And asexual reproductions, no meiosis in the cell cycle or no gamete. So in bacteria the asexual reproductions are taking place with binary fission, and in sexual reproductions there are different types of sexual reproductions like transformations, transductions and conjugations. So how binary fissions are take place in bacteria? So we are going to look upon that. And in binary fissions the bacterial cell actually is this, and it has some nuclide like nuclear body, because bacterial cell do not have any nucleus, so they have nuclide or nuclear body, and the nuclear body is attached with the mesosomes. So this is the structure of mesosomes. This is the cell wall of the bacteria, we all know that. So how this binary fission take place? So on the first stage of binary fissions the cell wall will not break, so the nuclear body is attached with the mesosomes. So the mesosomes start breaking down into two parts. So old mesosomes, this was the old mesosomes and this was the new mesosomes. So after dividing the mesosomes completely, and after dividing the mesosomes completely, the nuclear bodies start again dividing. So the nuclear body got divided, and there are two nuclear bodies. One is the main nuclear body and the another one is the daughter nuclear body. So after that the cell wall start dividing. dividing into two parts. So the cell will start making a depressions like this. So this is the mesosomes and this is the nuclear body. This is the mesosome and this is the nuclear body. And finally in binary fission the cell gets divided into two parts. This is mesosomes and this is the nuclear body like this. This is how the asexual reproductions or binary fission take place in bacterial cell. So let's talk about the sexual part of reproductions in bacteria. So the sexual reproduction in bacteria mainly taking place first with conjugation. Conjugation means to conjugate with one another cell. Like in conjugation, bacteria have two dimorphic gametes. We can say dimorphic. Gametes are bacteria like male and female bacteria. So the male bacteria actually here is donor bacteria and female cell is the receiver. So in conjugation, for example, this is the bacterial cell and this is the plasmid and this is the nucleus or nuclear body, and they have some structure like external structure which is called F pili. This is called F factor or fertility factor also. So the F pili start attaching with the female gametes like this. The F pili start attaching with the female gametes here, and they start making a conjugation between the two cells. This is the conjugations between the two cells. This is the nuclear body, and they start transferring this, making a copy of this, and start transferring this plasmid or F plasmid into the female. F plasmid or F factor into the female. This is how the conjugation takes place in bacteria. So let's talk about another one is transformation. Transformation was first discovered by Griffith with an experiment with a bacterial cell. So in transformation, the bacteria, this F plasmid or F factor is in the environment, and from the environment this factor is taken by the bacteria in transformation. So the third part is transduction. So how transduction takes place in bacteria? So transduction is also a type of sexual reproduction. So transduction is the carrier of the gamete is actually the virus or the bacteriophage. For example, if there is a cell, bacterial cell, which has their own DNA, A, B, C, D. For example, this is the bacterial DNA in the nuclei. It is infected by a bacteriophage, which is actually a virus. So the bacteriophage attached here on a bacterial cell, and it started transferring its genetic materials into the bacterial cell. So this is the bacteriophage, and it transferred its genetic materials into the bacterial cell. So this is the genetic material of bacterial cell, and it started breaking down the bacterial DNA just to use the bacterial machinery to reproduce themselves by the bacteriophage. So these bacterial DNA get divided into number of genes, A, B, C, D, for example. So they captured their the host genetic machinery and started making their own proteins, and the bacteriophage replicates in the host genome or in the bacterial cell. So after packaging, when this bacteriophage are finally completed their life cycle or completed their replications, and they start packaging. So in the packaging, a few genes of the host or the bacteria get packed into these bacteriophages. So after the bacteriophage again infected another bacterial cell, for example, this is the new bacterial cell, then the bacteriophage which has the packaging, which has some genes of the bacteria in their body or in their packaging, so they start infecting this bacteria again, and they transfer the genetic material which is of a bacteria, for example A or B gene, and they through this the bacteria reproduce and make two double copies. So this is what we call transduction. So here we have completed the bacterial reproductions. Thank you for watching.